This video updates a 2012 video called GEO2R Analyze GeoData. Both videos demonstrate how to use GEO2R to analyze gene expression data present in GEO. Now more than 99,000 studies are available for analysis with GEO2R. Plus, we've added two new features, the ability to analyze RNA-seq data and interactive visualization tools to help you explore the data. In this demonstration, I'm going to analyze the GEO series GSE 41586, which investigates gene expression in a human colon cancer cell line. There the cells are treated with 3 concentrations of the chemotherapy drug 5-aza-deoxycytidine. I'll begin the analysis by scrolling down and clicking the button Analyze with GEO2R. Before I run GEO2R, note the full instructions link near the top of the page. This documentation includes edit options and features that are not part of this video, and provides references for the software that forms the basis of the tool. And be sure to read the section detailing GEO2R's limitations and caveats. Going back to GEO2R, we see a list of the samples in the study, along with metadata extracted from the original records. The samples are grouped by the 3 treatment levels, 0, 5 micromolar, and 10 micromolar 5-aza. The first thing we need to do is to define the groups of samples that we want to compare. Click Define Groups and type a group name into the box. You can name the groups anything you want. I'll type Control, hit Enter, then Low Dose, Enter, and High Dose, Enter. Next, we need to assign the samples to each group. I'll click on a row and drag the cursor over the relevant samples. Then click the group name that they belong to, and repeat for each group. Note that GEO2R supports up to 10 groups. I can now run the analysis. Down the page, I'll click on the Analyze button. This performs a comparison with default settings, and after a brief wait, I see several visualization tools, and below those, a table of the top 250 differentially expressed genes, sorted by p-values. In the table, I can view the gene expression profile for a single gene by clicking on the row. For example, I'll click on the top row for gene ERV-FC1. And I see a chart where the red bars represent the expression level of the gene in each sample. In this case, we see that ERV-FC1 is more highly expressed in the low-dose group compared to the other 2 groups. You could also use the Profile Graph tab to explore a specific gene. Here you enter either the gene symbol or gene ID number, which you can get from the table, or by downloading an annotation file with this link, Download Gene Annotation. For example, if I want to see the graph for nitric oxide synthase 3, I enter either NOS3 or the ID 4846. Clicking on Set displays the graph. I'll now go back to the table of the top differentially expressed genes. I can edit this table by using the Select Columns feature. This opens a dialog box that lists all the available statistics and annotation columns. To save the full set of results, use the Download Full Table link to export the results as a tab-delimited file. Now let's take a look at the new visualization tools. We see 7 different plots for visualizing the data. Let's look first at the UMAP plot. Click on the plot to open a larger pop-up for better viewing. In general, samples clustered together are more closely related than those found farther apart. See the full instructions that we saw earlier for more explanation. For this series, we see that the samples cluster within each group, but the groups are not closely related to each other. If I want to look at the relationship between samples before running an analysis, click Analyze before assigning samples to groups, and the UMAP plot and box plot will be displayed. The plots outlined in green have interactive features. I'll click on the first plot on the left, the volcano plot, then on the Explore and Download button. Here red dots represent genes with increased expression, and blue with decreased expression. Data below the threshold is colored gray. Scrolling over points on the graph reveals gene information, the log2 fold change value, and p-value. Radio buttons at the bottom of the window allow you to toggle between comparisons, called contrasts here, and you can download a tab-separated file containing data for the selected contrast. You may want to change test parameters and run the analysis again. Use the Options tab to make changes to the p-value adjustment method, or significance level cutoff, or log2 fold change threshold. I'll lower the significance level cutoff to 0.001 and the log2 fold change threshold to 2. Then I'll click Reanalyze. Now some of the plots are revised, including the volcano plot. Finally, the RScript tab displays the script used to perform your last analysis. You may want to save this as a reference. That's all for this demonstration. If you have any questions or feedback, please email the GEO team, geo@ncbi.nlm.nih.gov, or use the Email GEO link at the top of the page.